Okay, so this is my title, Integrating Modeling Domains for the Patient Digital Twins. So what I'm going to tell a bit is what is the kind of challenges of building models of, let's say, biology or patients. And then I jump to one example, what we do, and that is in our EU project, where we try to make a patient digital twin for hypertrophic cardiomyopathy patients. But just very quickly, this is my group. So you can see we are doing cardiac muscles, neurons, X-ray tomography, optical imaging, so a lot of all kinds of things. Most of the work is actually on let's say, half of the work in cardiac and muscle and neural astrocytes are actually on modeling side. So we are developing computational models of the cells, but also tissues, function of the brain, trying to understand how the brain is functioning as well. And then we are doing a lot of tomography, and of course, both are something that also in tomography, the problem is really inverse problems. But I'm not touching imaging and this talk, I'm more looking at the, let's say, computational modeling in medicine. And if you think about how the medicine models are now, how the rules in medicine, how the diagnostics have been developed, is that there's kind of two models, and one is rats, another is humans, and the rules and ways to do is experimental work that has been then gathered all the data, and we look how the population reacts to certain drugs, or is there some adverse effects, how the drug is helping at least most of the patients, or some diagnostics based on population models. And now, of course, there's more data, there's more AI, so things are getting better. But I think many of us here are also doing all kinds of computational models. Then what is the issues with those? But first, thinking about what we have now. So one is, of course, we have all kinds of data from your mobile phone used to wearable devices, to new imaging methods. We have gene proteomics, all kinds of omics that can be measured from your blood or tissue samples. And of course, that is the idea that now this is a big data. Let's crunch it and do some AI. And of course, you can see on the right, one is one kind of important part of the whole thing is that we would like to get rid of the rat on the kind of chain. So one way to do that is to go for in vitro human cell cultures, and that is like an emerging field. Now it's called microphysiological systems because it's more than just cells. There are tissues or even a combination of tissues on looking at interaction of different organs as well in test tube. And of course, the fourth domain is really the computational models, and those have been around for a long, long time actually. If you think how we are treating cancer, how we are calculating how the cancer treatment is done in radiation therapy, that is actually based on really heavy computational models of the patient. So that is actually a digital twin that has been around for some time. But my view is that we actually need to look all these domains really to look for the personalized medicine, integrating different computational models, different data that we have from this kind of big data side on patient data, that is population data, of course, individual patient data, and then other models that we are using for understanding how the biology work or how the let's say some drugs effect and so on. So actually, this is my big kind of message for you guys. So we could stop now, but let's go a little bit forward. So of course, idea is that we get rid of not just rats, but these generic human. So we are not talking about average response to drug or average diagnostics, but really we should have a patient that we are treating, individual patient or really defined population where we can look how these models work. And I think this is something we all are part of that if you are building computational models on whatever field, and that is how the numerical models are integrating data. So it's, and not just data, but also all kinds of information we have, whether it's by prior knowledge of something, and they have physics, chemistry, laws of nature. Of course, mathematics is part of the story, and the data. And if you'd like to make an individual model of something, then we need to personalize this data, and that is then we have a digital twin. And hopefully these models provide us new knowledge. And if we can personalize it so that they are really kind of patient-centric models, then we could really think about how these can help individual patients. Of course, they can inform us a lot about basic mechanisms of how things work and how certain kind of, what are the effects of drugs, for example, in medicine. But if you'd like to really think about how the individual patient work, then we need to think about how to personalize, how to bring that personal data to this computational model. And of course, if you think about having this spanner or whatever it is in English, to model that, you need the geometry of the spanner, you need the material properties. Maybe it's a homogeneous, something like cast iron system or something else. So it's relatively easy to simulate. But if you think about biology, that is a bit more complicated. Of course, we can take certain kind of really narrow topic and then try to understand how that works and use that. So we don't necessarily need all these levels. But if you think you are looking like modeling of the operation or how you put some kind of material implant to replace the bone, that could be just based on geometry. But if you'd like to make a model where we try to understand how, let's say, drug effects are kind of affecting the patient, we should look actually from molecules onwards, because there's the effects are happening. Then it propagates to cells, organs, tissues, and so on. So there's a huge amount of systems that are intervened. There's a lot of control system in various levels, from gene regulation to how the cell works, how the cell electrophysiology works, how the cell energetic works, how the organ works, and how the human works. And of course, we can think that we need to make a model of your behavior. And then we need to look more like high-level sensors, your mobile phone use. But still, we could think about how to integrate with that some of the knowledge of the brain function, or even chemistry of the brain. So these are biological systems, and there's really complex. They can be complex. Of course, modeling, we try to understand the core function, not to take into account everything. So they are multi-scale. That is one quite challenging thing. And many times there's, of course, a lot of data, sometimes, not always, but that the data is sometimes really badly defined, or it's noisy, it's scarce, it's not continuous timelines. Including maybe only patient. We don't have a control patient or non-healthy data. Sometimes we don't actually know the biological function. There's still a lot of uncertainty how certain biology works. So this makes things a bit challenging sometimes on making this kind of biological or clinical related computational models, especially something that is related to clinics, because we should also prove to make it applicable for commercial use or clinical use. Okay, just a short bit of flavor. This is something we have done a lot. This is an old picture of our computational model of single cardiomyocytes. You can see there's a sodium-calcium exchanger. That is the round part of that, and there's the equation for it. So you can see there's a concentration of ions, there's a functions to opening and closing these and pump functions. And how these data is coming from is actually painstaking measurement of single cardiomyocytes with this kind of patch clamping electrode. But we can simulate the whole cardiomyocytes. We can look to electrophysiology. So this is action potential, normal, and one disease. We can have a contraction. We can look how the contraction is changing, or we can look how the certain drugs are changing here. And this is just example what we did already years ago, but I think it's still a nice idea, still using this idea that instead of how to make drug testing, you cannot have a single individual, you need populations. So we made population of these models, tens of thousands in the computer. And then using those tens of thousands computer models that are a bit different each, representing like individuals, we can actually test how certain drug effects, like this one channel blocker that is used for treating arrhythmias, or this long QT. And on the left, there's a normal effect that is expected. Some of the models actually start making arrhythmic responses, and some of the models, quite luckily, small number of the models died in a way. So no animals were harmed, no humans were harmed, but now we could go back and look these individual models and why certain models died, why certain group of individual might generate arrhythmias. What are they kind of constitutes of the models? So the idea, of course, if you go to clinics, it's not just looking what is happening in single cell, but to span this modeling two different scales, understanding the single cells, understanding the kind of insides of that, the molecular and cellular kind of, let's say, organs, then look at how tissue works, how the whole heart works, and finally, kind of thinking how the patient heart is functioning. And this is actually what we aim to do. We have one of these, of course, there's still question mark here, and then I'll show this picture again. We can discuss whether that's happening. But this is what we aim to do in this, our EU project that we are coordinating here in Tampere. It's called SMASH-HCM. And it's stratification management guidance of hypertrophic cardiomyopathy patient using hybrid digital twin solutions. Of course, that was the EU call, and we answered the call and got this kind of nice project.